So why don't we get started? So it's my pleasure to introduce Brenda, who's going to do her thesis defense today. Usually we give a little introduction to introduce the students and put things in context. Recently I'd been giving introductions where I just sort of said, here's Brenda, and that was it. But I had an idea for the way I wanted to describe Brenda's work, and so I decided to do more. And part of this is because I've been looking at other people introducing their students. So Denise will compare them to Disney princesses, and Justin always has a superhero, and Gary Nolan will, present a movie and things like that. But I saw an opportunity that there's, no one has used feats of strength and interpretive dance. Okay, so I'll do this. So what Brenda did is a great project that was actually accepted for publication last week at PLOS Biology. So that's that, and it's nice, and that's out of the way. What she did was, regular mouse immunology, which was a first for us. We've been a fly lab up until now, and Brenda was the first person to work on a mouse project, which ended up also turning into a human project. But what was really important is it's the first project in the lab that was really dependent on someone being able to code and look at things mathematically in a way that we haven't before. So she's opened up our lab to doing that kind of stuff. So I want to explain what she was doing. So one of the ways we like to look at systems is to find out how stretchy they are. Okay, so for example, this is a spring and a bungee cord. Okay, and so what I could do to demonstrate how elastic the cord is I can put an extra pound of pressure on it. You can see that the cord gets longer and longer, right? Until I would pull so far, and at some point, something's going to fail. And I won't do that because it's probably me that's going to end up failing and end up getting hurt. And so actually, Alex Louie, who's sitting here somewhere, did an experiment just like this using fruit flies, where he stretched the system and found out how it stretches. And we'd love to do this with people, but you can't because it's a combination of illegal and impossible. Okay? And Brenda will explain why. So she had to find another way of doing this, okay? And so this is what I want to explain. So imagine that, I want to put on a glove because I did this yesterday and I ended up with blisters all over me. So imagine this is a person, and they're going to represent a point in a multidimensional space, okay? And the auditorium is this multidimensional space. So it's easy to see 3 dimensions. You've got X, Y, and Z, but there are other dimensions as well. You can have the dimension of light, you can have the dimension of sound and smell, and a whole variety of different dimensions. So as I think about this space, you can imagine that this is what you would do is you would look at it and say, I noticed that it exceeded the space a little bit. It would be hard to do things, say more. So Brenda did assumption is a simple kind of path. The path she showed was this one here. So she wants something, she's going to look at things that are just circles that form loops. It's okay, it's not going to hurt you. Okay, so she makes loops, and the idea is to measure the properties of this loop, and she'll show you how she does this with data. And the tricky thing she did was that she was able to figure out how to make these loops, not just for animals that are sick, but that are animals that are going to die too. Okay, and so here we do it in multiple dimensions now. So what she's doing is she gets the ones that are in red and shows they exceed a simple space. Okay, and so that's what she's going to show you, but with
also showing us that in cases where we might not have those parameters available, we can look at others. So for the first step, can we create these disease maps? Here's an example where we show that we can. So the next question that I had is, can we create these maps when time is unknown? So here I'm representing a mouse where we've taken all the samples for the full course of the infection. But what if there's cases where we can only take data from every single mouse, so it's an individual mouse for each, or if we're looking at human data? And as David mentioned, it would be very unethical to try to follow someone, not give them any form of treatment. So we needed to find a way to see if we could create these maps looking at cross-sectional data. And so one of the ways that David has used to sort of help understand how we came about as to creating our maps using these this type of data is that here is a snapshot of what individuals heading towards Tressiter, or, you know, they're heading in different directions, right? And so the samples that we have, you know, it could be all these individuals seen here in this image. But there's properties for each of these individuals. They're unique, they're diverse. And so when we're trying to understand where they're going during disease, we can then look for specific properties that can tell us a little bit more about the direction they're heading. So for example, these individuals, they would be grouped together, clustered together, because they're heading, it's obvious they're heading in one direction. So what we did is we used nearest neighbor analysis, and that name makes a lot of sense, right? So you're going to make connections with your nearest neighbor. And so what it does is it looks at the similarities between samples and then makes a connection. So here I'm showing you the mouse data with knowing when the samples were taken. And then what I did is I took that information out. So this is what it would look like if it was cross-sectional. And then I applied the nearest neighbor algorithm. And this is what we see. We can reconstruct these maps using only cross-sectional data. So this is really exciting for us because it showed that we could take this very diverse data and still be able to create the maps that we were interested in. And so I wanted to look at human data. So what I did is I took the human malaria data set. And what this data set has is that it has samples for 96 malaria-infected children, and then 61 H-match control subjects. And again, they were only sampled once. So to help understand what we were seeing with the human data, I just want to show you again the mouse data. Again, this is taken every single day of the infection. So it's a very nice loop. And if people were wondering why I was showing a donut for my flyer, this is why. So just sort of indicating to you that it's a very nice loop. We have all the information. We have our complete donut. For the human data, someone's taken that piece of our donut. And so here's what the human data looks like. Here I've colored controls in red and then infected in blue. And so what we're seeing is that we're really getting a lot of great data when when samples are being taken when kids are sick, but we're sort of missing this area of recovery. So it allows us to see that there's another way of, that maybe we could look at the data by collecting more around the different stages of the infection. And here's just a 3D movie showing you, how we're not really having samples in this region of the infection. And so if I could get some help with the lights. No? Oh, okay, never mind. So for the next couple of slides that I'll be showing you, we came into contact with a group in Menlo Park It was a startup that was created by a math group here at Stanford, and what they do is they look at the shape of the data. So we were really excited to take our data and use their software and see what is the shape that we see. And this is what we get. So without any indication of when those samples were collected, the mouse malaria data forms this loop-shaped curve. And what I'm doing here is I'm plotting, I'm coloring based on days post-infection. And so here's the early stages of the infection and then later stages. And so indicating to us that maybe by day 25, they're starting to look more like their healthy selves. And I won't spend too much time on this slide, but just trying to show that we do have strong evidence to support that these loops are very common throughout the data. And we also looked at other additional data sets. So in the first row, what I'm showing is data that's all mouse data. So here is what I showed you before. Here's mouse malaria looking at cytokines. Here's looking at mouse data where they're infected with influenza. And then on the bottom row, I'm showing that human data set that I was working with. This is what it would look like using the software. And I can talk about the colorings later, but just the big picture here is they're making that loop. This one was really exciting, clinical data showing humans or volunteers that decided to be infected with malaria, and they collected clinical data, and it's longitudinal, and then also the space that we see when we're looking at the vaginal microbiota. So what the question that I had, can we create these maps without time when time is unknown? And the answer is yes. So the next question was, can disease maps be applied to study resilience? And just as a brief reminder, seeing if individuals are heading a different direction or are they coping with the infection a lot easier than others. So with the Plasmodium chabauti model, 20% of these mice don't survive the infection. So the same idea, same concept, but then collected samples for mice that didn't survive the infection. And just to show the space that we were really interested in is this one where we see the dying mice, represented here by the thicker lines, they're occupying a completely different space here than the mice that recover from the infection. And just to briefly mention, reticulocytes, we're using it, there are newly formed red blood cells. So indicating to us that red blood cells are being created to replenish the loss of those red blood cells that was occurring. So this is all nice. This is great. This was exciting. But we have to find a way to find statistical information from our maps. So with the help of our collaborator, Ann Thomas Tate, we ended up finding a method to do that. So here I'm representing our maps a little bit different. The live mice are in orange, dead mice are in blue. And so the idea was, okay, so these maps, they're circular, forming, so it's a circle, and we have samples around this circle. And so if we center it and polar transform the data, then we can get an angle and a radius for each of these points. And by doing that, and this included the live and dead data, we can then look at the angle and the radius. And because we're interested in this space where the dead mice are heading to having a different trajectory, we can look at that specific area and then do statistical analysis. Here I'm showing you if we measure the radius for live and dead mice, there's a statistical difference. And then we could also do logistic regression analysis where here on the y-axis is the probability of being alive, and then radius is on the x-axis. And so the probability of you surviving decreases as you move further away from the center. So you have a longer radius. And the reason why this works so well is because you have time and angle have a really strong correlation. So can our disease maps be applied to study resilience? And the answer is yes. So we can take our maps, we can see when there's bifurcations, and then do statistical analysis on these maps as well. One of the other things that we mentioned in our paper is that we wanted to see how genetic variation affects resilience. And the thing that I did is I took, the human data set also collected information about mutations that could occur in this individual. So people can develop, can obtain sickle cell trait if they let me backtrack a little bit. So red blood cells, they have a protein called hemoglobin. If there's a mutation in the hemoglobin, then not as much oxygen can be carried. If you have one mutation, you're noted as AS. If you're normal, it's AA. And then if you inherit both mutations, then it's SS. So what I'll be showing you here is this is the human data, red blood cells again by reticulocytes. If I look for only the individuals who have a normal red blood cell, then they seem to be making a bigger loop. For the individuals who have a sickle cell trait, which is thought to help fight malaria, is that it seems to be forming a smaller loop. And so indicating to us that maybe one of their future steps would be to identify SNPs that are associated with changes in resilience. So for the conclusions for the first part of my talk, and I've noted here 1 through 4, the different things that I discussed, and they are that 1, disease maps are useful in describing host dynamics of infections, as we can see here by the different types of parameters that we can use. We also can order that cross-sectional data. So we might not know if a person comes into the clinic, we don't know when they got infected. With doing nearest neighbor analysis and creating our maps, we can better identify what stages of the infection they might be in. We can also do statistical analysis, and then we can also start thinking ahead and thinking about ways that we can look at how genetic differences affects how we fight off an infection. So for the remainder of my talk, I want to talk a little bit of the stuff that I'm doing now. So I just want to preface with it's very preliminary. I hope it generates a lot of hypotheses and questions, and I'll try to answer as many as I can, and happy to talk to you guys afterwards about it too. So there's this data set that looks at the vaginal microbiota where women self-sampled for 70 days. And we have samples, or this group has samples for 125 volunteers coming from the University of Alabama at Birmingham. And they also recorded their hygiene practices and also sexual activity for each of these days. So it's a rich data set. And one of the questions, so we learned about this data set after going to a conference and talking to Larry Forney at the University of Idaho, who studies the vaginal microbiota. And he wanted to know, is there a better way of visualizing the data just to maybe obtain more information from? So in the paper that I cited before, this is one of the images that they have. And so on the y-axis is phylotype relative abundance. This is over time. And then the phylotype are colored in different colors for different phylotype. And then all that extra information is down here below. So there's a lot of information here. And so we were also interested in seeing if by using the software provided by YASD, can we better understand other information from this? Can we obtain more information from this data set? So here's the YASD maps that are created when I take those samples, and this is only using 35 women. And what I'm using here down below is I collected or I'm showing you examples of the most dominant microbiota that is associated with a healthy vaginal microbiota. So here's Lactobacillus crispatus. Here's just the samples that you'll find that have the abundancy is high for Lactobacillus crispatus. For Lactobacillus inners, they're located in this space. And then for Gardnerella vaginalis, they're located in this bottom region. But I was more interested in seeing how individual subjects' microbial composition changed over time. We're looking at 70 days. So how do they move around this space? So with Ayazdi, here, the only information that I can obtain is where the samples are located. So most of the samples noted here is where those samplers are located, but I don't know when day 1 starts or when the study ends. So what I decided to do is to take these Ayazdi maps and then find the angle for each sample. So then I know where they are in that space. And then, so this is an example of what I should be seeing is that if this is one subject, maybe she moves in this direction, and then maybe she moves back in the other direction. So it's bidirectional. There is no unidirectional for this space. So it might be difficult to sort of see those changes if I plotted them in this manner. So what I additionally did is that I took time and put that on the radius. And I'll go over how these maps are going to look like. So here, again, these are the bacteria that we know are dominant species in the vaginal microbiota. And so darker regions is where most of the samples are located, around this space for Lactobacillus crispatus, around this region for Lactobacillus inners, and again, Gardnerella vaginalis at the bottom. So very similar to what we were seeing with our Yazdi maps. And then the exciting part is, here's where I just highlighted one individual. So this individual, this is day 0, this is day 70. They all stay, or she stays, and her microbial composition stays in this region of that map. Here's another subject. She stays in this other region, but it seems to be very similar to the previous subject. And then you have individuals where they sort of sample the space a little bit more. And then you have others who sort of jump, right? So this individual was around this, was in this space and then moved onto this other region of the plot. And then there's women who just might be going all around they're going in all space, different directions. I hope it just is illustrated here by this image. So I'm really excited about this. We also know, as I mentioned before, there's records of the different disturbances. So I'm trying to, so the observations that are made is that the women have a similar microbial composition over time, and then could these changes in the microbial composition correlate with the patterns of disturbance? Haven't done that yet, but maybe we can learn a lot about what it means to be, we don't know if this could be referring to stability, if they're very stable. Does that make them more resistant or more resilient against perturbances, or maybe being less stable helps them do that. So for the final conclusions, just showing you the images of what I focused on today, but I just hope that I leave you eager with wanting to create your own disease maps. If not, please come and talk to me. I'm happy to look at the data, and also eager to hear more about the progress of what I'm currently looking at, and that is the vaginal microbiota. So with that, I'll just brief acknowledgements for now. Just want to acknowledge the current members of the lab. Thank you so much for helping me through these last couple of weeks. And then former members of the lab, as well as the collaborators that we had, and then my thesis committee as well, and Erin Mordecai for being my chair today, and funding sources, of course. Thank you. And I'll take any questions. Oh, yes, Denise.
You showed us a lot of mouse data in was that multiple doses, or was it a dose at the ID50, LD50, and how does that change your disease maps when you use it?
Right. So the data that I showed you here today was only using 10 to the 5th infected red blood, so 1 dose. We have done in the past different dosages, and what we noticed is that the maps, we only did this one, so I just we would have to do it again, but we did see changes in the map, in the disease map. So with a lower dose, if I'm correct, it was a smaller disease map, and then as you increase the dose. But the thing that we did notice is that no matter what, I think like 10 to the 2 and on, you all reached the same parasite load. So they all went to that extreme, independent of what the dose was.
So what is the LD?
The LD50 would be, I'm trying to think, because we did 10 to the 5th, and so that's 20%. So I'm thinking somewhere around that area, yeah. Mike.
So with a lot of these kind of trying to find the trajectory in a multidimensional space
Yes.
kind of analysis, knowing distance is usually important, and knowing that distance on one axis and distance on another axis are similar. So for instance, when you're making a circle, if you get that wrong, it's going to be less than a line. How do you figure out how much of a change in granulocytes matches a certain change in blood cells or whatever for the scaling of the different?
Oh, so you're talking about how that linear relationship that we saw, could it be because of changes in the scaling?
Well, so when you have the plot with the circle,
Yeah.
how do you know what scale to put the x and y axis on, basically, to make that circle?
I see what you mean. I don't think it's a problem, because we're using that just to help us map where people are. So we just want to find something that can create a loop, and then in the future, and we have people working on it right now, where you take a sample that was not, it was done before or after, and you try to see where they are on the map. So, oh, no. Yes.
Yeah, lovely. Have you tried taking just clinical data from patients that, they collect tons and tons of temperatures and red blood cells and white blood cells, and then asking your program to find the loops? And that's one question. And then the other is whether these loops, which type of loop gives you the most information in terms of where people are? Because I imagine some of them have no correlation depending on the variables that you choose, and some will be tiny and some will be very broad, and
Right.
which are the nicest loops?
Yeah. So that is something that we were observing as we were looking for the best disease map. And so we know that there's work being done in our lab that has done that. So we have a system where you can automatically just put in your data set, it'll find the disease maps for you, and then also can find whether, because it has a training set and then another set of data that you can use to then see where they will map, and it has a high predictive value. So I'm hoping that with that we can have a better understanding if we're finding the right spaces. Sarah.
Really interesting stuff, Brenda. I just had a question to make sure I was really understanding all of your newer data correctly.
Yes.
You first generated your loops for the changes in the vaginal microbiota, what was it that you used to create those loops? Was it PCA plots? Was it just a bunch of the line?
Right. Yeah, I didn't mention it. So it was, we used the metric cosine. So what cosine does is that it looks at the angle between samples. I know it's sort of like we used the angle of the samples in that Ayazdi space, but then you also can use it as a metric. So with cosine, you're only interested in whether there's collinearity and not necessarily the differences in magnitude. So that Ayazdi map for the vaginal microbiota, it's grouping the samples together based on the similarity between them, the collinearity between them. Cool.
I noticed in some of your disease maps there are smaller circles that were up sort of toward the healthier side of the graph. Maybe in particular in the second part of the talk.
Which, do you remember? Oh, yes, okay, yes. I'm gonna like flip through if you see.
The pretty colored ones that you.
Oh, yes, okay. Say no more, say no more. Okay.
So I see 3 circles.
Ah, yes, okay.
So I was wondering.
These. Okay.
Do those signify anything important? And maybe do you think there are circles involved in health as well as in disease?
Yeah, so the region where we are seeing those other 2 circles appear in this map is where you see that species Lactobacillus crispatus. And so crispatus is really known to represent a healthy vaginal microbiota. And so what I think is happening is that there's also records of the menstrual cycle. So when the menstrual cycle is happening, crispatus leaves the environment, and then Lactobacillus inners sort of temporarily takes that space. So I don't know if that's sort of what's the reason why we're seeing that extra loop, because maybe, you know, it comes in, it comes out. I don't know. So just from what I know of the information, I'm suspecting that that's what's causing those different loops to also appear. And then loops between health and loops and with disease. With health, I don't think you'll create loops unless you know things change while you're healthy, and then you identify those things that change over time. And then if you find a slight shift, then I think you can create a map. Peter.
Yeah, so to construct these graphs, it seems like the markers you're using for disease, that those can be sort of arbitrary, as long as it correlates somehow with, and is temporally shifted away from the health problem. And so you're going in, looking at things that you'd expect to change over disease, so like immunological markers.
Yes.
I was wondering, I don't know if this would work or make sense, but could you use this in a forward discovery sense, where you go through and in an unbiased manner survey a lot of different parameters and see which ones of those form loops over time? Yeah. And then go through and like have a sense that the ones that are forming loops might have something to do with the disease, even if they aren't at first glance directly affiliated with the immunological response.
So maybe like thinking about like a disease that's very asymptomatic, but then there's things that are changing but aren't physiologically, nothing that you see that indicates that there's something wrong, but that you could use these maps to say, okay, there's something that's happening. I think maybe looking at understanding what's the baseline health. We can look at immune cell types and then understand what is the baseline of all these cell types that indicate that you're healthy. And then if we are seeing changes in those individuals, maybe we can see that. I'll think about it. Maybe there could be a way that we could do that. I like it. Seth.
Really excited around the development of the vaginal microbiota. And I was really particularly struck when you said that it doesn't necessarily have to be something that comes from your blood, but you can monitor your weight. And with the advent of all this connected self and Fitbit data and all this. I was wondering how you see these particular graphs scaling to the type of data that we have on self-monitored data.
Yeah, I think I think they should think about it and read my paper about it, because I mean, no, that's where we're heading. That's what we want, right? So, the Fitbit tracks sleeping cycles, and so if we see indications to us that something's wrong with our maps, then that would be great. I'm trying to think of a scenario. Yeah. So when you get sick, you might be sleeping more, and so then you'll be occupying a different space between sleep and non-sleep. Yeah. Yes, but we want that to happen. We want to use that type of data with our maps. Amanda. Yeah. Yes.
Or like predictors of where someone would be in certain places. Can you pick out the best way?
Yeah. So we really, for this model, we really think seeing a replenishment of the red blood cells is a really good marker. So seeing reticulocytes appearing during the infection is important. Yes, you can find those markers because you see that they are very good at separating the data. How I showed you the non-survivors and survivors, really distinct spaces, but I think you need two, right? Because as I mentioned before, when you have one parameter, you don't know if they're getting sick or recovering, so I would say you would at least need two parameters to do it. Two good parameters. So red blood cells and reticulocytes for our model.
What sort of disease space do you see this model really applying to? It seems like right now it's the case of an infectious disease in which you are bound to get sick and then recover, or get sick and then die. But then there are chronic diseases, the ones with really different manifestations. So I'm imagining your donut would be much thicker, that sort of thing.
Yeah. So that's the exciting part too, right? That you're right, we only started an acute infection, right? But it would be good to study chronic infection, what sort of maps those are creating. We think it's sort of going to be like a spiral, right? So for people who might have autoimmunity, and there's, days where everything's okay. So, and then you have what's it called? A flare, and then suppresses flare. So you can create maps with different types of diseases. And that's the great thing about it, right? You can pick whichever one you're interested in.
Okay. You can go to your next.
Oh, I forgot. Oh. Started on a different slide. All right, so I just want to do some brief personal acknowledgements. So I was going to start off, I was thinking, maybe I can find a donut for each person in my life. But I decided not to do that. But the only one that really stuck out was this one. I thought this was David because he always wears his green North Face jacket. But it would be a really shame if I didn't describe David and my relationship using disease map. So we all know who the pathogen is. I'm just kidding. He's really a mutualist. And what I'm doing here is monitoring my stress levels. So I'm really excited. I joined the lab. me and Jose are working on these mouse models. We see David occasionally, so things are working. And then the data comes, and David gets really excited. And so then my stress levels go really up, because I was really nervous because sometimes I thought, oh, I can't do this. What am I going to do? But David helped me realize that I can do it, so my stress levels went down. And then as I transition towards moving out of the lab, I know that there's going to be less of David around. And so that makes me stressful because I've always been working with him. But hopefully we continue our relationship and you can still guide me and advise me even when I'm not in the lab. And so stress levels will go down. And then here's just an image that really describes David. He's fun and goofy, and I love Halloween because he participates every single time. And then I also saw this book and I thought of you. My committee, thank you so much. Right now I'm feeling really excited. This is probably how I feel inside. And it's all because of your guidance and making sure that I stay in the right path. To my lab, thank you so much, guys, for being my lab mates. I love that we can hang out together even outside of the lab. We travel together. We travel to Costa Rica, and we share really special moments together. And then to the current lab, and then a special thanks to Poonam. I didn't have a picture of you, but here she is. Thank you for being there. David was not there on Thursday, and so I was panicking, and Poonam gave me my pep talk, so thank you. And then a special thanks, of course, to Jose for being there during the early stages of the project. Thank you. And then my immunology group, you guys are awesome. I can't see all of you, but I see some of you. And you guys are great. I love hanging out with you guys and having birthdays together. I especially like some of you more. And lucky for me, there's a bunch of new immunologists that I've gotten to know over the years that I love more than others. A special thanks to the group above. You guys are some of my closest friends, and so thank you so much for just being that group of really close friends. And then to the DARE cohort, you guys, we share this journey together. A lot of people are interested in working with or applying to DARE, and so I get really excited. I cannot stress enough to apply. It's a great group. You should meet all of them. Meet everybody and be part of the group. A special thanks to BioAIMS. I've been involved with BioAIMS all the time. And a special thanks to Terrence and Summer for just making graduate school 10 times better. And then also Anita, because she doesn't take me seriously, so that's always good. And she always laughs at my jokes, so that's nice. To my kids from the SSRP program. Okay, to my friends, thank you for being there. They're actually live. There's a live feed, so thank you to the people who couldn't be here, and a special thanks to Godson. So I have a joke for this, so I think I can do it. So I think you copied me. Yeah, I got my PhD in immunology. You're getting your PhD in computer science. But I got my PhD first. So I win. And so, and if you get your PhD next year, biology just takes a longer time. And then, to the love of my life, peanut butter. I'm just kidding. You know who I was. There's a joke here too. This is when we were hot. We've grown bigger together. Hopefully, we can grow skinny together again. Parents, you guys are great. Thank you. Thank you for all your love and support. I love that you love me. And then, sorry. Let's do that again. And then, and then you occasionally love Cesar. And with that, I'd like to conclude. So thank you.
So now we meet in private with Brenda for about an hour or so, and then following that, outside of the Fairchild building, there'll be champagne and donuts.
Yay! I didn't go.